hands up.
Okay.
Hi, so the study we did for our presentation was maternal single nucleotide polymorphisms in the fatty acid desaturase 1 and 2 coding regions and how they modify the impact of prenatal supplementation with DHA on birth weight.
So the hypothesis of the study is twofold in a way. The first part of it is that single nucleotide polymorphisms, or SNPs, which we'll be referring to them as throughout the rest of the presentation, in the fatty acid desaturase genes, which are the FADS genes, alter the activity of the delta desaturase complex, resulting in heterogeneity of LC polyunsaturated fatty acid long chain polyunsaturated fatty acid concentrations among individuals. And that hypothesis was created based on previous studies regarding SNPs in the FADS genes. And for this particular study, the hypothesis is that maternal SNPs in the FADS genes modify the effect of prenatal DHA supplementation on birth weight. So in 3 long-chain polyunsaturated fatty acids, or DHA, is essential for neurodevelopment and the regulation of gene expression during early development through the effects on stem cell proliferation. The way that DHA affects fetal development has been examined in several studies, and 2 of the main suggestions for why it's important is that it increases the duration of gestation and that it increases blood concentrations of insulin-like growth factor, which is why it's presumed that it can affect child birth weight. And the availability of long-chain polyunsaturated fatty acids during the infant's first months of life is determined by their mother's dietary intake and how they metabolize these fatty acids. So previous studies have examined how prenatal DHA supplementation affects birth weight, and they found either no conclusive results or heterogeneous results, meaning that the DHA concentrations in the infants has been different, and they haven't been able to figure out why as of right now. So the fatty acid desaturase genes, or FADS, in the specific study, they examined FADS 1, 2, and 3. The function of these genes is to regulate the desaturation of fatty acids by introducing double bonds between defined carbons on the fatty acyl chains. So essentially, they convert omega-3 and omega-6 essential dietary fatty acids into the DHA and AA long-chain polyunsaturated fatty acid derivatives that are used in the body. So the single nucleotide polymorphisms in these genes have been suggested to be responsible for the heterogeneity in the results of prenatal DHA supplementation. And they found some evidence that these polymorphisms on the FADS1 and FADS2 genes affect the activity of the delta desaturase complex, which is why they have seen different results in concentration of LC polyunsaturated fatty acids in the mother and infants. And there's also been some studies that suggest that SNPs in the FADS3 gene can also play a role in the metabolism of DHA.
So this study was a double-blinded randomized controlled trial. There were originally 1,836 eligible women, but after the inclusion material was examined, 74 women did not meet it, and then 668 declined to participate, leaving 1,094 women who were randomly assigned to either the intervention or control group. The intervention group received 400 milligrams of DHA per day at 18 to 22 weeks gestation, while the control group received a placebo at 18 to 22 weeks gestation. The inclusion criteria was women between 18 to 37 years of age who plan to deliver at the Mexico Institute for Social Security General Hospital. And they also had to breastfeed for up to 3 months and live in the area for at least 2 years after delivery. So informed consent was used for genetic testing and was given to 654 of the participants. They used anthropometric measures to after up to 24 hours after delivery, and then the birth weight was converted to Z-scores. They took fasting venous blood, and it was split into plasma, buffy coat, and erythrocytes. And then genetic analysis was conducted from the extracted DNA in the buffy coat. We'll talk a little bit more about that in the next slide, but a subsample of 140 women with genetic information also had information on plasma concentration on long-chain polyunsaturated fatty acids at baseline. And then finally, maternal dietary intake was assessed with the use of a 110-item food frequency questionnaire. So 4 single nucleotide polymorphism clusters resulted from the test map of LD, which we'll talk a little bit more later. And then the SNPs were as followed: the RS 174455, RS 174556, RS 174602, and RS 498793 were selected as tag SNPs representing the fatty acid desaturase genes, the FADS1, FADS2, and FADS3 gene clusters. These 4 tag SNPs were included for the assessment of the effect modification of prenatal DHA on birth weight and for the sub-analysis of plasma concentrations of AA and DHA. These 15 SNPs were selected due to previous evidence regarding their effects on long-chain polyunsaturated fatty acid metabolism.
For the statistical analysis, first they used chi-square test, t-test, and ANOVA to test for differences between the groups. And they also used a proc univariate procedure in the SAS 9.2 software to look at the DHA and AA concentrations to see if they were normal. They also used an ANOVA to assess the differences in weight for age, as Tori mentioned, to get the Z-scores across the intervention groups of the FADS SNP genotypes. When it came to choosing which 4 SNPs they wanted to use for the study, they used the Java linkage disequilibrium plotter, and this calculated the allele frequencies, Hardy-Weinberg equilibrium, and linkage disequilibrium, and it plots them graphically. And so from this, they chose one SNP randomly from each cluster with an LD of above 0.8 to represent each cluster of the 15. So that's how they chose the 4 SNPs that Tori mentioned in the methods. They used a linear regression analysis to assess for any association between the FAD SNPs and plasma concentrations of the arachidonic acid and DHA. And first, the 4 SNPs were looked at separately, and then after, they used a multivariate logistic regression on all 4. And then throughout all of their analysis, a p-value of less than 0.05 was considered significant. And then to account for any type 1 error, they used the Bonferroni correction. Now for the results of the study, first, what they did to just make sure that the groups were consistent, they looked at the maternal characteristics, including age, socioeconomic status, years of schooling, intelligence test, height, BMI, their first pregnancy, and dietary intake, and that was at baseline. And then for the offspring at birth, they looked at gestational age, length, weight, and head circumference. and they found no significant differences. When it comes to the actual results of the study, they first they found that the RS 1744-55, RS 1745-56, and the RS 1746-02 were positively associated with arachidonic acid plasma concentrations. And you can see that right here. And then for the SNP RS 1745-56, that was the only SNP that showed a positive association with DHA. And with a p-value of less than 0.05, they found that RS 1746-02 was inversely associated with the DHA concentration. And then for the results on DHA and prenatal weight, down on this left bottom picture, you can see the SNP for RS 1746-02, and you can see it has a p-value of 0.01, which shows that they found a significant heterogeneity with that SNP. They found that the offspring of carriers of alleles TT and TC in the intervention group were heavier than those in the placebo group. And then they found no differences in the offspring of this RS 1746-02 CC homozygote. And for the rest, the three other SNPs they looked at, they did not find any significant heterogeneity.
So there needs to be further research conducted on the biological mechanism behind the interaction between the minor allele, FADS2, SMP, RS 1746-02, and prenatal DHA, and the subsequent effect on birth weight. This is because in the study, in carriers of the minor allele of FADS2, SMP, RS 1746-02, women who received the prenatal DHA gave birth to significantly heavier offspring than those who received the placebo, but this difference was not observed in carriers of the major allele. So that's why the interaction between the minor allele and the prenatal DHA should be researched further. So each SNP's gene location gave variance to the nature of associations between the plasma long chain polyunsaturated fatty acids, AA and DHA. It also was evident that the maternal FADS genotype does play a pivotal role in birth outcomes, along with long chain polyunsaturated fatty acid metabolism and the availability for the mother's offspring. So some things that the overall variance in how we saw the different impacts of DHA supplementation on birth weight that we saw in the results could be explained by the contrasting responses to the prenatal DHA of the target population, in this case, the Hispanic women. And this could either be explained by their individual genetic makeup or their polyunsaturated fatty acid dietary intake. So some strengths and limitations of the study. The strengths were, these are the important strengths, were that the data collection and the laboratory analysis, they were all done within a clinical setting, and they were conducted by trained personnel, and they were both the collection and the analysis protocols were all standardized and validated by these trained professionals. Additionally, participants were part of a population with a high dietary intake of the N6 fatty acids. And they also had a high prevalence of the FADS genotypes, which is associated with the slow conversion of long chain polyunsaturated fatty acids. Even though this demographic was a more restricted subsample, it still held the characteristics that were similar to those of a full study population, which still made it a strong study. Finally, the last strength was that the hypothesis was able to be tested within a randomized controlled trial with high quality information, like I just mentioned, on birth outcomes and a wide range of maternal and offspring sociodemographic characteristics. So finally, a few of the limitations that were noted from this study include that there was a proportion of the women in the original study that didn't provide consent for genetic testing. It was only a small number of the original. Additionally, there was information on plasma concentrations of long-chain polyunsaturated fatty acids at baseline that were only available for a subsample of 140 women. And lastly, the total fatty acids were measured in plasma. They were not measured in erythrocytes. Unfortunately, at the time of this clinical intervention, this was the method that was available in Mexico. So they were not able to conduct those erythrocyte measurements for the fatty acids. Going into future research from this study, overall, it really just goes to show the importance of integrating and studying the current advances in the field of genetics and how we can implement those into future nutrition interventions. They really go hand in hand. Another future study could be studying the mechanisms of action behind the association between AA and DHA with different FADS SNPs, and then see if these results would be consistent across other populations, as other populations have different diets and a different genetic makeup than the population studied here. Some more future research would be to assess if the impact of DHA supplementation, what impact it has on birth weight, and even assess if there's long-term child growth effects and outcomes, specifically in mothers who carry the minor allele that we mentioned earlier. And finally, conducting future research in the differences in DHA and polyunsaturated fatty acids intake in trials assessing genetic makeup of the target group and birth weight outcomes in other populations. And then these are our references. Thank you so much for listening.